Hey, thank you so much for having me and flying me up here from Boston. It's really a pleasure to be here. And I spent a lot of time in Canada. My wife actually did her graduate work at McGill and actually is up in the Canadian Arctic a lot for her own research. So we've traveled up here a lot, but I've never made it to Halifax. And so it's great to be here, and thank you for having me. So I've given several versions of this talk, and in the past I've often focused more on the basic science. What is DNA? What is RNA? What is protein? Partially because we get to the end of like an educational session, and people would come up and be like, well, this is great. We've had all this stuff for all day, but could you answer one question for me? What's a gene? It's like, that's a great question. And it's a really good, legitimate question. What is a gene, right? And this is really important. We keep talking about these mutations and genes and pathways. We kind of have to start with the basics. So for a long time I did that talk. I'm trying out something new. We're going to instead really try to focus on therapy and this idea of personalized medicine and what it is we're looking for when we're developing new cancer therapies, how we're thinking about personalized medicine both in general, but specifically how we're starting to bring this into WM. So hopefully there'll be some time at the end for questions, and I'll be sticking around. Hopefully, weather permitting, I'll also be at the walk. So plenty of opportunities also to ask questions then as well. So I work at the Dana-Farber Cancer Institute at the Bing Center for Waldenstrom's. Now we are a center devoted exclusively to Waldenstrom's, and we literally see thousands of Waldenstrom's patients every year. It's a full clinic that supports 2 doctors, a dedicated nurse. We have multiple clinical trials going on. And also, for people, just so people are aware, most of the patients we see, we just, it's just to consult. I mean, to see that many patients, even in the United States, honestly, a lot of them are coming in from California. They're not going to come down for every infusion. They're coming down to check in, get a sense of what's going on, what insights, second opinions. And that's really how we envision WM care, just as like what I think we heard earlier in the question and answer center about what's going on in Canada. You have regional centers like here where then people can get referred to for more specialist treatment, because in a rare disease, that's really, really important. And so the big picture of the Bing Center, and I just want to mention this because I'm the first person to come up here who's not actually an MD. So if you have a medical question, I'm really not the right person to ask. We have a wonderful expert right here. The Bing Center is led on the medical side by Jorge Castillo and Steve Trion, who founded the center. And then the sort of basic genomic discovery and signaling is run by me and by my colleague Guang Yang, who then takes some of that signaling work, clarifies it, and actually does drug development. So you'll hear a little bit about some of the drug development testing work that's going on there. That feeds back in the clinic. They come back, they tell us, hey, this generated a whole bunch of new questions, and it sort of forms this really great series of interactions. So for starters, what is cancer? Right? Really basic. For the most part, cancer is caused by accidental changes in our DNA. And we're going to talk about what that is over the next few minutes. And, as these changes accumulate, your body, certain of these instructions that we call genes, they lose their function, or sometimes they gain new functions. And if enough of these accumulate, some of these cells will start to forget a little bit of their own programming, forget about what they're supposed to be doing, and they'll start to grow uncontrollably. And that is what we call cancer. Now, what makes cancer so tricky, right, is cancer is really derived from yourself. It's your own cells. Right? And so when we're talking about targeted therapy, right, we are trying to reach in and attack a cell that came from you initially, right? And just hit that cancer cell. And this is really tricky, and this is the essence of cancer therapy and of targeted therapy in general. Can you identify elements of the cancer that are unique to that cancer, or at least more targeted towards that cancer, so we can hit that hard and not hurt the rest of your body, or at least limit the number of cells that get hurt. So we're going to talk a quick bit about the history of cancer therapy and how it's developed. And actually this really got started actually in Boston by Sidney Farber, who was treating children with childhood leukemias. And this is back at a time when really the only option was surgery. And when you have a blood cancer, surgery is not going to go well. And he, in the lab, had noticed that there was this folate dependency for some of these tumors, and he had this idea, well, maybe we could use a chemical to act as a folate inhibitor that might have a specific effect on the tumor. And it did. And this was a huge deal, right? You could give a chemical, and it would target just the tumor, well, not just the tumor cells, but it would target the tumor cells, and it would have an effect. And it was a huge, just mind-bending change for everybody. So, also going on, in a completely unrelated parallel track, were the World Wars. Now, why am I going here? During World War I, unfortunately, chemical warfare was routinely practiced and was a really horrible thing. And mustard gas, in particular, was used extensively. And so in the space between the wars, there was a lot of research that went into what is mustard gas, how does it work? Can we come up with ways of preventing it? Can we modify it? Most of this was secret government research at the time. But there was no real practical application that came out of this until World War II. And there was a tragic accident, actually, in Bari, Italy. The Allies were invading Italy, and the Germans came through and bombed a series of boats just outside the harbor. Now, one of those boats was carrying mustard gas and a bunch of ammunition. The whole thing exploded, it went everywhere, and there was widespread exposure to mustard gas, which was not a good thing. Treatment of these people, however, they flew in a bunch of US doctors, and they were following these patients, and one of the things we noticed was, okay, lots of things going wrong, but you know what? Their white blood cell count is consistently going down, right? And that's interesting, right? Even with people who survived and recovered, their white blood cell really crashed, at least initially. And so they started thinking, we learned all this stuff studying mustard gas. Maybe we can modify its components and use this for therapy to attack specifically the immune system. And actually, there's a whole class of almost most of the chemotherapies that we use today are actually derivatives of mustard gas, including, for instance, chlorambucil. And they're great drugs, and they're not related at this point to, chemical warfare agents, but that is their origin. And so what we now understand is how they work, which is that they damage the DNA, and cells that are actively dividing are going to be more sensitive to DNA damage. And so when you think about a chemotherapy, and you think about what the side effects are, well, for starters, tumor cells are growing pretty fast. That's the whole, we had this whole definition of cancer. They're dividing out of control. Yeah, so it targets them. But that's not the only thing in your body that's growing, right? And if you think about it, well, your skin is being replaced all the time. And that includes your internal GI tract. So you get a lot of toxicities there, right? With the skin, with the GI tract, and your blood and bone marrow, the healthy part. You're replacing your immune system, your blood all the time. Again, a lot of these agents that damage the DNA, and this includes radiotherapy, but also chemotherapy, are going to be hitting all dividing cells. So on one hand, it's targeted. We're able to identify something unique to cancer, rapidly dividing cells, figure out a way to hurt them, but there's still a lot of bystanders. It's better than previous options by a long shot. These are really great therapies, but we'd like to think we can take it one step more. And in fact, over the years, a number of increasingly targeted agents have been discovered. So rituximab, which we've heard a lot about, is a compound that actually is using the same kind of molecules that your body produces to go after infections, right? So, we're always told, like, you get the chickenpox once, we hope. And you get all these diseases, ideally, again, once is how vaccines work. And it's actually you produce these antibodies. And this is one of the ways that your body remembers and can fight infections it's seen before. Well, we can make artificial ones to go after individual targets, and targets that we think are more specific to the cancer cells. So CD20 is something that is on immune cells, specifically on B cells, and it's a great target. You also, unfortunately, deplete out healthy B cells. But, again, this is better than, for instance, causing DNA damage throughout your whole body, so this is a good first step. You also, now in clinical trials, we have a CD38 antibody. It's the same idea. It was developed actually for multiple myeloma. It's in clinical trials at the Dana-Farber right now, and we're very excited about it. You have proteasome inhibitors, which sort of target the way that proteins and parts of your cell get recycled, and that can put stress on cells. And it's a particular kind of stress that we've discovered that cancer cells are particularly susceptible to, without going into any of the details. And it's actually worked incredibly well. And then sort of newer agents, we have immunomodulatory therapies, which in all honesty have not worked great, like thalidomide and lenalidomide. And new stuff that's really cutting edge, like the PD-1, PD-L1, and the CAR T cells, vaccine therapies, that are coming online, which honestly have not really been looked at in WM. Now, a lot of these things are really exciting. You see stuff in the papers, everyone gets really excited. They are really exciting. They're maybe not as exciting as the papers make them out to be. We're seeing some amazing responses in select people, but we're still trying to figure out where they work, what some of the long-term toxicities are. It's a less straightforward intervention at this point, though it's something that we're really excited about, how we can basically harness the immune system to reactivate and fight cancer, because your immune system really is its own kind of targeted therapy that can learn how to fight a particular thing and is incredibly efficient if it can recognize it. So all the things I've been talking about, right, are things that are just true for whole big classes of diseases. And in reality, each patient is a little different. And the more we're studying them, a lot of these diseases that we used to think of as one thing, actually we now think of as several different things. And arguably, there's diseases like diffuse large B-cell lymphoma, which we still call it DLBCL, but in reality, we don't even treat the subgroups remotely the same way anymore. And so if we can understand what the underlying biology is, can we use this to develop new therapies, to figure out which therapies are going to work best for an individual person, and start tailoring our choice of therapies based on individual characteristics. So the genetics of a tumor cell is really, well, it's my focus, and for a lot of personalized medicine, it's really our launching point to try to understand how these different types exist, what the biology of the disease is. And so I want to, before diving in there, just talk a little bit about the basics of genetics and how we think about it. And, genetics and DNA in particular is a loaded term. Especially if people remember in the late 80s and early 90s, there were these twin studies that came out where people would always talk about wow, the twins separated at birth, they like the same kind of chocolate bar, they use the same toothpaste, they name their dog the same thing. And, it turns out if you get two random people and you go through enough stuff, you're going to get some interesting connections. And that's not to say that identical twins are not really, really similar. They are. But actually, the long-term studies that have been done on this are noticeable not just for the similarities, but also for the differences. And really, one of the things that we've learned is that DNA is really not, they used to say things like, there's DNA for determining whether you're a dog person or a cat person, right? Whether you like coffee or tea. And no, you don't. No, there's not. There's not. it's DNA is one part of who you are, right? And that's really what I want everyone to understand, is it's a blueprint for teaching your body how to do things, right? It's not a blueprint for you, right? Again, identical twins don't grow up to be identical people, right? They have the same set of tools, but they go in different directions. So, all right, most people are completely unique, and this is why you can do crime scene investigation stuff on TV and unique identifications. But in reality, we're all pretty similar. And we need some way, however, of talking about these variations in common. And this was actually the real special thing about the Human Genome Project. It was this massive effort. And it gave us a common base, right? The Human Genome Project, interestingly, is not a person. The genome has never existed and will never exist. It's actually an amalgamation of a bunch of people in Albany, New York. Really, randomly chosen, and any individual stretch is one person, but actually the next stretch is from someone else, and they're not linked. However, it gives us a universal reference, right? So even if that reference turns out to be wildly uncommon, which definitely happens, we can still talk about it. It's like, okay, this is the baseline, and then this is how everyone varies from the baseline, which is why some of us are tall, go prematurely gray. Any of these things, like we can start talking about how these differences arise, and try to understand how DNA may or may not play a role in that. Now, there are over 3 billion base pairs in the human genome. Yeah. There are over 3 billion base pairs in the human genome. And you inherit one full copy from each parent. And you have this inside every single cell in your body. And you can measure this, right? I mean, this is a lot of information. You can actually string it all out. And it's a long piece of information, right? It would kind of make sense. Look around. People are complicated, right? I mean, heck, mice are complicated. Worms are complicated. A single cell is really, really complicated. But think about that amount of information stuffed into every single cell in your body. And it's not just in the cell, it's actually in this little tiny compartment inside the cell called the nucleus. So, as amazing as this is, right, this is still a lot of information. And actually, it's so much information, it can't all be accessible. And this, in fact, answers a bunch of long-standing questions we had in biology, which is, if you inherit the same, this DNA from both parents, and it's the same DNA throughout your body, there are some complicated exceptions to this, but for the most part, I'm pretty sure that my eyes behave pretty differently from my liver, from my blood and bone marrow system, from my heart. They all act really differently. I can expose them to the same thing. They're going to act differently. They certainly developed into very different types of organs. If they have the same DNA, how the heck did that happen, right? Well, actually, they may have the same DNA, but they don't have the same set of instructions. And it's because there's so much. It's wound up so tightly that any individual cell actually only gets to see a small amount of it. And it's a combination of chemical annotation to silence them, or literally the packing and unpacking of it, that means that your heart sees a different set of genes than your liver does, right? Makes sense when you stop and think about it, right? Now, here's the problem, right? Is it's all still there, which means, okay, so maybe, for instance, your B cells are supposed to see a certain set of genes. That can be undone, right? And it happens all the time in cancer. So things that aren't supposed to be available become available. Things that are supposed to be available suddenly get silenced. And it can start to alter the program. So we were talking about mutations, but it's not just mutation. Actually, it's this whole larger picture of how genes get regulated, how the DNA is made available or unavailable, that works as a whole to manufacture a cell and also can go wrong and cause cancer. So to put it all together, I would really like you to think of the DNA as being the operating system for a cell. And I would like a show of hands. You have been sitting all day. We need to get some activity here. So how many people have a personal computer? All right, pretty much everybody. So how many PC users do we have? All right, Mac users? All right. Any Unix people? No? Okay. Everyone who raised their hands with PC, right? All of you, assuming you keep your operating system updated, the DNA of your computer, right, the operating system, is identical. They are all identical twins of each other. All the Mac people, your computers are all identical twins. None of you use your computers in the same way, right? What's on your computers, how you use them, what they do is completely unrelated, right? And that is the key about DNA. It is giving a set of tools, right? And those tools are really important, right? Try switching between Mac and PC and you'll get confused. I mean, they're real tools, right? But it's about this interaction with the environment, right? So you have DNA, and DNA itself doesn't do much. It sends instructions out into the cells. You don't want to bring your one copy of something and have it run around and do all the work. No, you make a copy of it, of just the relevant piece, and you send it out as an instruction, right? You need more of it, you send more copies out. And that gets translated into protein. And protein is out there and does actually most of the physical work in the cell, helps with the signaling, but this can go back and it can bind to the DNA, and it can help activate transcription or this copying. So it helps activate, turn genes on and off. It can help control this winding and unwinding, which genes are available, right? And so the protein there is interacting constantly with the environment. So cells are interacting with cells next door, right? And it's those interactions together that actually will form a tissue, and that tissue interacts together to form organs, right? Organs interact together to form full systems, right? And all together, all these levels of interactions happening simultaneously is what makes us all possible. So it's that interaction is what makes everything tick. It's not the DNA. The DNA is just like your operating system. It just sits there. It's how it gets used. It's that interaction that's really important. And when we think about what's going on in cancer, we really need to think about these interactions. We're going to come back to this later. So before we go any further, it was once presented to me, and it's really the best way of describing the way mutations work that I've ever seen. And so I'm just going to steal it totally from a Canadian, Martin Krasinski at the BC Cancer Agency. And I would like you guys to read it back to me. So do you think you can do that? All right, let's do it.
And if we accumulate too many of them, our abilities is overwhelmed.
And that really is what happens, right, at the cellular level. So what's the most common spelling mistake in Waldenstrom's macroglobulinemia? It's this mutation, this single of the 3 billion base pairs. There is a base pair that goes wrong in a gene called MYD88. And so what is MYD88 anyway? Your immune system is comprised of two parts. There's the smart, intelligent, adaptive part, and that's the part that learns how to fight a particular disease, why you don't get the same disease multiple times, makes antibodies, like we talked about previously. And then there's a not so smart part. And it's also really critical too, because actually it takes a long time to learn how to fight something. And if we depended only on the smart part, we'd be dead long before we were able to mount a response. So we need something that is actually fast, doesn't really know what's going on, but it's like, hey, I'm pretty sure I saw a virus, pretty sure I saw a bacteria. Let's just fight stuff. and without a lot of knowledge about any specifics. And in fact, that is where MYD88 fits into the picture. So there's these proteins, they're called the toll-like receptor proteins, and they literally are designed to pick up things common in bacteria and viruses. And if they get triggered, this body knows, hey, I'm not supposed to see this. This is not supposed to be there. Let's let the rest of the immune system know that there's a foreign invader here. Let's send out a big inflammatory signal. Let's get the parts of the immune system which are really good at finding foreign things and breaking them down. Let's get them all here. And more importantly, let's grow and survive to fight this thing. Right? And so what the mutation does is it causes a conformational change in the protein that makes it independent from the receptors. So it will now be active all by itself. It no longer needs to encounter a bacteria or virus. It can just signal all on its own. And what this does is it sends this signal all the way down, and do not worry about the names, right? This is, concentrate on the big picture here. But it sends the signal down through the ARX, through into this process, this whole process called nuclear factor kappa B, or NF-kappa B. And this is a critical factor for immune cells, for B cells, and it is a very pro-inflammatory signal. And it's part of, in the normal immune system, what will go and be like, hey, let's send out the message that we've seen something bad. We want to have everyone come over and fight this, and heck, I'm going to fight this, right? I'm going to grow, I'm going to survive, and I'm going to fight this infection. And in fact, you can see all this activated NF-kappa B as green dots in the center of the cells. And here we have stained the nucleus blue. So that's where all the DNA is. And you can see all those green dots are there in the blue parts of the cells. If we put a MYD88 inhibitor in, unfortunately, we do not know how to do this with real live people. We can only do this with cells. But when we do it, actually, all that NF-kappa B gets kicked right out of the nucleus, and it's no longer active. Right? And the cells die. So this is really what's driving the vast majority of Waldenstrom's macroglobulinemia. Now, let's start to accelerate things a little bit here. Guang Yang and our group looked at this and was studying this pathway. He's like, I'm just not convinced that this is really the full story. And he did some investigation, and he realized that, well, what was known in the literature was true. There was another molecule that was at play. It was called BTK. And you guys might have heard of this. This is a target of ibrutinib. And it turns out in Waldenstrom's that independent of the IRAK pathway, MYD88 can activate BTK. And it also signals down into NF-kappa B. And so when we're talking about ibrutinib therapy, we have not figured out how to directly attack MYD88 yet, though we are trying. We can target BTK, and by silencing BTK, we can shut the vast majority of this signaling down. So I've talked about this one mutation, this L265P mutation in MYD88 that everybody almost has in Waldenstrom's. But this brought us to a question, is this really the only one? Right? Are there other possibilities? Because actually, in related to them, MYD88 is not unique to Waldenstrom's. And in other lymphomas, we see other mutations. What I can tell you right now, we just finished a 600-patient retrospective study on this. And pretty much, if you have a MYD88 mutation, 99% of the time, it's an L265P mutation. You may also have something else in combination with it, but 99% of the time, 0.7% of WM overall has one of these non-MYD88 mutations, or non-L265P mutations that are not, without having an additional L265P. And we actually learned this, because we had initially observed these two, actually when we were studying ibrutinib, because we noticed that all the people who didn't have a MYD88 mutation didn't respond that well to ibrutinib, except for two people who did really, really well. And we're like, what's different about them? We went back, we looked into MYD88, and then sure enough, there these guys were. And those were the two. And which brings me to a point. right? So 0.7% might sound like not very much, and it's not very much. But if you're thinking about a therapy with ibrutinib and you do test as not having the L265P mutation, it is actually important to get the full gene screened, because this does happen. In other diseases, it's much more common, but in Waldenstrom's it happens too, that there are these alternate mutations that will not be picked up by a typical test. And so it's just something to keep in the back of your mind. And on a similar note, not all, and this is what's confusing, both the scientists, like myself, and to patients and to clinicians, is that not all the tests are equally sensitive. And so one of the things that we're struggling with as a community is how can we make sure that the mediate testing that we're reporting includes some information about our level of confidence. I can tell you some of them are incredibly sensitive. Other ones we've had false negative rates as high as 27%, which means that we're giving bad information a fairly high percentage of the time. It's kind of all over the place right now, and we don't have a good solution for it, because actually the most sensitive testing really isn't possible in most places or practical. So as a community, one of the things we talked about this most recent workshop is how can we make this more available, cheaper, and at least communicate more accurately about it to ourselves, to you, the patients, so we can really have a common language for describing this. Now, the second thing I'd like to describe is the CXCR4 mutations. And unlike MYD88, we see lots of stuff here. But what you'll see is this, that text block, which I've been told looks like a bunch of computer gobbledygook by a friend of mine, is actually the protein code. So each of those letters is an amino acid, and that codes for what CXCR4 looks like at the protein level, and there's a sort of simulation of it right there. The DNA is there on the bottom, or at least the tail of it. And those boxes are where we see mutations. And you'll notice it's all at the end, right? We don't see them in the beginning, we don't see them in the middle, we just see them at the end. And so this is important because actually the end, interesting, is kind of a little bit dispensable for signaling, right? CXCR4 is a receptor. It's actually sort of a homing receptor for cells. It helps them figure out where they need to go. And they'll follow sources of inflammation, and they'll also, in particular, home to the bone marrow. Might sound familiar. It sticks through the cell surface and then signals downstream through a sort of internal docking ports you can see there in the bottom. But at the end of the protein, you'll see where the two arrows are two big handles out here, right? This is a regulatory region. So when the receptor is activated, it changes, there's some chemical changes that happen to these regulatory tails. And that allows the cell quite literally to grab a hold of them, sort of like handlebars, right? And pull it back inside. And this is actually how we turn the receptor off again. So you grab a hold of the handlebars, you bring it back inside, and you can stop the signaling. So what do all these mutations do? They cut off the handlebars. And that's exactly what happens. Without the handlebars, the cell can't pull it back in, and it'll just sit in the cell surface and just signal and signal and signal. You'll sometimes see these referred to as WHIM syndrome-like mutations. This is because there's actually something we'd call germline or inherited syndrome that's very rare. It's unrelated to WM. It's called WHIM syndrome, or Wart's hypogammaglobulinemia infection and myelokathexis. If you don't know what myelokathexis is, they invented it for the disease. It's actually has to do with the retention of certain kinds of immune cells in the bone marrow, which is what happens. Because with this homing signal on constitutively, these people are basically normal, except that almost their entire immune system, and particularly their neutrophils, are stuck in the bone marrow and won't come out. And so we actually have to put in CXCR4 inhibitors, and then actually their immune systems come right back out again, and they're fine. Until it wears off, and then they go back. So we've actually been working a lot with these people because it's the same set of mutations that we see. And so we're learning from each other about how we can develop therapies. We'll talk about that in a little bit. So we see these CXCR4 mutations primarily in the context of patients with MYD88 mutations. And so we can break them up into four basic categories. People who don't have either mutation, sort of wild type, double wild type Waldenstrom's, no MYD88, no CXCR4. And these people have the lowest levels of bone marrow, the lowest levels of serum IgM. You have patients who have MYD88, which we kind of treat as the baseline. It's the largest population. We have two different kinds of CXCR4 mutations. So there's two kinds of mutations we can think about. One of them is a sort of a scrambling mutation. It doesn't truncate it, but it just scrambles everything in the tail, so it's no longer recognizable to the cell. And ones that literally just chop it off. And the ones that just chop it off, we see this much higher rates of higher levels of bone marrow involvement, higher levels of serum IgM, more serum hyperviscosity syndromes. They're more symptomatic. And regardless of what kind of CXCR4 mutation we see a lot fewer instances of splenomegaly. So big spleens, also lymph node involvement tends to be much rarer in this group. And if you think about it, you have a mutation which is causing the cells to home to the bone marrow. So not surprisingly, they're homing to the bone marrow instead of the lymph nodes. The other thing we see with this group is therapeutic resistance, particularly to ibrutinib, but to a number of other things as well. And so this is something we're starting to track a little bit more carefully, and this is where we start to get back to personalized medicine, right? By tracking things like MYD88 and CXCR4, we can start to incorporate this testing into clinical trials to understand who's really going to respond best to this therapy. If we know in advance someone doesn't respond well to a particular therapy, why on earth would we ever give it to them, right? So this is where we're trying to get to with this. And indeed, ibrutinib is one of these therapies. This is a breakdown of ibrutinib response. So there's a percentage of reduction of IgM. So the lower is the greater level of reduction on the right and with the blue. And you can see those people who were wild type for CXCR4 really had much bigger reductions than those people with CXCR4. And as I mentioned, the CXCR4 people took an extra 6 months to get to there, right? Whereas the wild type people got there in a matter of weeks. So, and the people who had neither mutation really didn't respond, right? You see the same pattern in reverse with hemoglobin. So I've made the CXCR4 group sound pretty scary. It turns out it doesn't matter that much. It matters in terms of your response to therapy and some symptoms, and I think ultimately it will matter quite a bit in terms of management and the choices we make. However, in terms of overall survival, we don't actually see that big of a difference. Where we do see a big difference is in people who don't have either one, the sort of double wild type group. Actually, they're the ones where we see have higher rates of mortality, higher rates of something called transformation, which is where Waldenstrom's itself gains enough mutations and becomes an aggressive lymphoma. So it goes from an indolent lymphoma to becoming an aggressive lymphoma. And again, this can be really not a good situation. And is a major driver of actually the reduced survival of this group. So we literally just completed the study. I sent in the proofs last week on these sort of MYD88 wild-type Waldenstrom's patients. And please do not write down all these genes. You don't need to memorize all of them. It's not all that important, other than to say that these are genes that are associated with non-Hodgkin's lymphoma. If you were to go through and ask what genes are associated with non-Hodgkin's lymphoma, this would be your list. And a lot of them are targeting NF-kappa B. A whole lot of them. And what's important is, if you remember, NF-kappa B was the sort of end target for MYD88. If you look at this chain here, they're all below BTK. So you have all these activating mutations underneath BTK. So we can inhibit BTK all day, and it's not going to matter to these people. And this, for us, was our first real scientific explanation of why we were getting the observations we were with the ibrutinib therapy in Waldenstrom's. So ibrutinib is a great drug for people who have MYD8 mutations. For those who do not, we need another approach, and it's something that we're working on. Now, another thing I study is not just the DNA, but I also study how the DNA is used. So we talked a little bit about gene usage and transcription. That's when individual pieces of DNA are copied, like blueprint copies. I mean, if you can imagine, in the olden days, building a cathedral, this is a massive undertaking, right? There are hundreds of thousands of things to do. There's statues, there's woodwork, there's masonry, right? It took a lot of work to get those initial blueprints out. You're not going to make copies of all of them and give them to all the artisans, and not to mention, they're going to be completely overwhelmed, right? You're going to copy the relevant part, and you're going to send it to the mason and be like, this is what we need. And the stone carver, you're going to be, hey, we need like 25 gargoyles. Just, can you do this for us? Like, this is specs. But you're just going to give them that part. You're going to make a small copy out and give them that part. And that's what the DNA does. And so we can track gene usage by looking at how these copies are being made. And one of the things we notice about Waldenstrom's is that some of these genes are being used in ways that are abnormal. And one of the pathways in particular is one that regulates cell survival. Because under normal circumstances, our cells are being damaged all the time. Physically, right? You get punched, you play hockey, you know, you're going to get some physical damage, but also chemically, right? Through radiation as well, through what we eat, through the exposures we have. Oxygen even, turns out, can cause problems. So we are accumulating damage all the time. And what will happen is the cell will make a call at some point and be like, you know what, I've accumulated so much damage, it's better for me to be replaced by another cell than to try to fix things. And so there's this whole survival pathway that regulates this response. And it's called the BCL2 family. And not surprisingly, cancer cells like to abuse this. So they overexpress the pro-survival, and they really sort of silence out the pro-death signaling. Now, the good news is that there are drugs for this. And so we just finished a clinical trial of venetoclax, which is a BCL2 inhibitor, which has been very successful in other related lymphomas. And it went really well. We're going to be reporting this out at the American Society of Hematology in December. And again, ultimately, we hope to start bringing it into combination therapy, ultimately. But it targets this pathway in WM. So let's put this all together, right? It's not too much, right? So there's a lot going on. But by understanding the biology, we can start to rationally develop new targets and also start studying how these different types of WM respond differently to each of these therapies. And because of this, we have things that target BTK. And that's not just ibrutinib. We have a next generation of inhibitors that are coming through, like acalabrutinib, zanubrutinib, even more coming down the pike. We have venetoclax, I had just mentioned, that targets the cell survival. In clinical trial at Dana-Farber right now, we have an antibody trial of ulacupamab, which is an anti-CXCR4 antibody that silences CXCR4 signaling. And so we're using this in combination with ibrutinib to see if we can get CXCR4 wild-type-like responses in CXCR4 mutant patients. So it's actively enrolling right now. And in development, we have IRAK inhibitors. So we didn't talk about it too much, but we've talked about how successful BTK is, but MYD88 also signals down through these IRAKs in the same pathway. And we've just been pretending like that's fine. So we're really, and while it is a minor part of the signaling, it is still very real. And so we're looking in the combination therapies with IRAK inhibitors and BTK inhibitors to see if more fully silencing MYD88 will make a difference. And also in development that we're inheriting a little bit from the myeloma program is ERK inhibitors. And this is important not just for CXCR4 signaling, but also for downstream signaling from MYD88 itself and its relevance to actually to BTK resistance. So when we have people who are resistant to ibrutinib, we see a lot of upregulation of the MAP-ERK pathway. We're going to talk about that in a minute. So So we're now trying to bring some of these new drugs that are being tested in multiple myeloma into the WM clinic. We just got approval to do a very small phase 1, and hopefully we'll be able to have some feedback on that in the next couple of years. So we've been through a lot, and there's a lot to take in. I just want to cover a few more things quickly, and these are just highlights from the international workshop that just happened in New York City, and from people who are studying genetics from different parts of the world, heavily biased by the people who are friends of mine and were willing to give me their slides. So with that caveat, and these are their slides, so I'm going to try to highlight individual things. Don't worry if not all of it makes entire sense. Some of it doesn't make sense to me either. So one of the big themes is tumor evolution. So we've talked about all these mutations, but in reality, they don't just all happen at once. They don't, tumors don't spring fully formed into existence. They slowly accumulate mutations, and you hear about MGUS, right? People can live for years with MGUS, and nothing happens, right? And then it becomes, at some point it grows out and becomes asymptomatic smoldering WM, which can go for years with nothing happening. And then it becomes symptomatic. What happened? Why is that, right? We don't really know, but one of the things we do know is that we see an accumulation of mutations as you move down this chain. And so we want to try to understand how these mutations stack together, how they interact with each other, and use that information to see if we can come up with new targeted therapies and new ways to attack WM. So the first thing I want to talk about is deletions in 6q by a good friend of mine, Maria Luisa Guerrera. And 6q deletions, we didn't talk about it. It's one of the really common things in WM. It can happen in, up to 40% of patients, I would say. And all this is saying is that your DNA is actually broken up into 23 distinct pieces. It's not one big super chain. And so if you've ever heard of the company 23andMe, that's what they're referring to. It's the 23 individual pieces, or chromosomes as they're called, that make up the human genome. And so 6 is just the 6th biggest one. And it turns out that they kind of look like propellers, and so you can divide them into 2 pieces. The P arm, which is usually short, and the Q arm, which is long. And what we see is deletions in that sort of latter Q arm. The DNA just gets taken out and erased. It's gone. And sometimes it's only a few cells, and sometimes you look at the tumor and it's all the cells. They're all missing it. And we've always wondered, what is this doing? Why is it there? And it's a big region, lots of genes. So I had done a study that initially identified a bunch of genes which were abnormally represented in deletions there. And so Maria took a couple of these and decided to follow up with a sort of more sensitive study. And one of the things that she found was that people who had deletions that really encompassed all the cells, right? So if you had a gene that was deleted in pretty much the entire tumor, it wasn't just that gene, it was all of 6. And you can see it in that big blue cluster up there. So the darker the blue, the more cells are affected by that deletion. And there's 2 interesting things about that group, right? The first thing is that we see that happen in people who are asymptomatic, who have smoldering Wallenstroms, and people who have symptomatic disease. See it in both cases. Where we do not see it is in people who have CXCR4 mutations. That was a surprise. We also don't see it in people who have mediate wild-type WM, either, for that matter. Now, one of the things that we did then is we took genes that were uniquely expressed or dysregulated in the context of 6q deletions, and just tried to visualize graphically and cluster them together and say, okay, let's just take all WM patients, take this gene set, and let's just see what happens. How do these samples relate to each other? And typically what you would expect is, all the people with deletions in 6q are going to be on one side of the graph, and everyone else is going to be on the other side of the graph, because we selected genes that do this, right? Now, every once in a while when you do this, you get something weird, and that's what this is. So we do see this, everyone on one side who's deleted, everyone on the other side, but there's this you can think of it like a north-south group. There's this big green-blue axis in the bottom there. That's CXCR4 mutated patients. We didn't choose any genes related to CXCR4. So that was weird. Why are we getting this clustering based on these genes? And we actually went back and we checked, and in fact, there was a gene set that was being regulated in an identical fashion in both groups. So they're mutually exclusive of each other. You either get one or you get the other, but they regulate the same set of genes. And why is this important? So going back to this idea of MGUS, Smouldering, and active WM, we already know that MYD88 is an MGUS. CXCR4, we see an MGUS. We just discovered we see the 6q deletions in Smouldering WM, we see an active WM, and through some really wonderful studies that actually happened in Spain and in Italy, really dissecting out the MGUS clone, what we can say is we don't see them in MGUS. So we really think that the CXCR4 and 6q are playing some role here in that transition from MGUS to Smouldering, and that this set of genes that they're co-regulating is really giving us a guide as to what genes are responsible. And this can, again, help us learn new targets that we can go after, because if the cells are still dependent on this pathway, maybe we can block them and we can knock the thing all the way back to MGUS. That's not a cure, but that's pretty good. I'd be happy with that. So one other thing, just very briefly, we talked about transformation. Why do some people transform? This is Cristina Jimenez from Spain, who actually just joined our group and is now researching ibrutinib resistance with myself. And DLBCL is a very aggressive, complex disease, and we really don't know why this happens. And so she did this big study tracking patients who didn't transform and those who did, and trying to understand what was different, but most importantly, trying to understand the pattern of these things. So not all mutations wind up making it to the clone that ultimately transforms. And so can we identify mutational patterns that occur in people who do transform? And can we identify the mutation that actually flips the switch, right? Where you were Waldenstrom's before this, and then after that, not so much. And so that's really what she worked on. And the biggest thing that she discovered was that it was people who had mutations in a gene called CD79B that were at a much higher risk for transformation. Now, to be clear, not everyone who had this mutation transformed, but a very high percentage of them did. Also, she observed people who lacked mediated mutations, like we had, also transformed, though she was not able to identify any particular cause. So this is our first clue about how this works. CD79B is, in fact, very commonly observed in diffuse large B-cell lymphoma, as is MYD88, though at a lower rate than it is in WM. Another thing is by my friend and colleague Guang Yang. We literally sit back to back. And his big work has been discovering that it's not just enough to activate NF-kappa B. We have to understand what NF-kappa B does. And to better explain this, I just want to point something out. And I like to remind all of my research colleagues of this, which is, and this is true not just for WM, this is true for the indolent lymphomas in general, multiple myeloma, CLL. We can't cure it, right? We do not know how to cure or to kill these cells in patients. You knew that already. We do not know how to keep the cells alive outside of a patient. We can't. We cannot figure this out. Yeah. So for all this fancy genomic stuff, there's a lot we still don't understand. And the reason for this is that these cells are not really dependent on their environment and their interactions with the environment. And until we better understand that, we're not going to be able to make progress. And what Guang had figured out was that I talked about how this innate immune system signaling that sends out these warning messages. We see bacteria. We need to activate inflammation. Just go, go, go. Well, that doesn't just impact WM cells. It's your whole body, right? Your whole body goes into this sort of like, I have an active infection going on. And in turn, that provides a bunch of signals back to the tumor cells that they desperately need. And so, and that in turn activates a molecule called HCK. And HCK, believe it or not, can activate BTK. And it turns out it's the major source of BTK signaling. It also activates ERK and AKT and other things that are very, very critical to cell survival. And in fact, the big pro-survival role of MYD88 is creating this pro-inflammatory environment that activates HCK. And if we block HCK, these cells die. I mean, they don't like BTK inhibition. They're gone with HCK inhibition. There aren't any great drugs right now. You know, ibrutinib is a weak HCK inhibitor. It's one of the unique things about ibrutinib, and one of the reasons we think it works as well as it does. But one of the things that we're doing right now is we are actually developing HCK inhibitors at the Bing Center with the Harvard Medicinal Chemistry Group. And so there's some really exciting stuff. We're working to partner with some pharmaceutical companies to see if they can pick it up, because we're not a pharmaceutical company. But we want to see if we can get it far enough that we can convince them this is a real thing and it's going to make a difference for patients. So this also applies to ibrutinib resistance, because one of the things we discovered, and this is really amazing, is in people who become ibrutinib resistant, a certain percentage of them get mutations in BTK. And it's right where ibrutinib binds to BTK. So that makes sense, right? You change the place where it binds, it can't recognize it anymore, it doesn't work. Okay. But here's the interesting thing. It's only a small fraction of the cells. And this is not just WM, this is other malignancies as well. And what Guang was able to show was actually what was happening is that this small percentage of cells was reactivating this inflammatory pathway. And once that was activated, everybody's resistant to ibrutinib. And in fact, he could put a small set of mutant cells in a plate, cover it up such the cells couldn't get through, but the message could, put more cells that didn't have the BTK mutation on top and treat the whole thing with ibrutinib, and they were all resistant. So this is really a very critical thing. It was a big deal. So we still need to better understand the microenvironment and how this works, but this is a really important breakthrough, and I really want to give Guang a lot of credit for uncovering this. So hopefully we'll have more information on this in coming years. And then last of all, I want to talk about something that is really important to this group, because this is a study that, that CURE, the IWMF Canada, is funding. This is the WM mouse. And so this is Ruben Carrasco, presented at the meeting. And what he did is he took human MYD88 and he introduced it into a mouse. Now, there's a couple of things that are worth mentioning here, which is that the MYD88 mutation is just in your B cells, just in your Waldenstrom cells, right? So we can't have the mouse have MYD88 everywhere, right? That's not really going to recapitulate anything. So what we do is we actually put a little bit of a blocker in front of the human MYD88 so it won't work and won't be expressed. But if it's exposed to a certain chemical, it will cut itself out. That blocker will cut itself out and it will reactivate the gene. So what he did is he made a mouse with this blocker in it, with MYD88, and he made another mouse that expressed this chemical, but only when in the presence of if a B cell program was turned on. So genes that were specific to B cells, if they got activated, it would turn on this chemical, it would go, it would hit the MYD88 blocker, cut it out, and boom, all the B cells and only the B cells would suddenly have this human MYD88 mutation in them. Pretty clever. So at first, we didn't really observe too much, right? What we did observe is a bunch of the mice who had this mutation, and it's not the prettiest thing to look at, had these rashes right here on their cheeks, right? Loss of hair, it's actually pretty bad. And some of them actually had to be put down because of it. And we just didn't know what to make of it. And then just this year, there was a case that got published of a young girl who had a spontaneous mutation in MYD88. And so this happens sometimes. You can't copy 3 billion base pairs perfectly every single time. And so when people are born, all of us have some mutations in us that aren't from our parents. It's just unique to us. And unfortunately for her, hers was in MYD88. And so she had juvenile arthritis. And then at some point, lo and behold, she developed a neck rash, just like the mice. And so when this paper came out, all of a sudden we were like, oh. And we started looking at the mice, and sure enough, they had these inflammatory signals. And we looked a little closer, and what we learned was they actually had a lymphoproliferative disorder that was related to the rash, but also was present throughout their body. Was not cancerous, so it's not one cell, but there was too many immune cells, and they were overproducing IgM. The IgM was much, much higher. And finally, in some of the older mice, 3 of them actually developed real bona fide lymphomas. And in particular, something that's worth noting is that these mice started secreting something called CXCL13. Now, this is just another chemical signal that cells make, but it's one that we've associated with Waldenstrom's. As a matter of fact, it correlates very strongly, both in terms of expression, but also in terms of the amount in the blood, with the bone marrow burden in Waldenstrom's. Likewise, it corresponds with the level of hemoglobin suppression. So this is really pretty exciting. We see elevated CXCL13. We see, albeit at a low rate, some lymphomas. So the question is, we're clearly on the right track. Mediate by itself is not enough. How can we take this to the next level? And so the person who happened to be working on this study with Ruben was none other than Maria Luisa Guerrera, who did the 6Q study. So they decided that they were going to go and try to make a 6Q deleted mouse and cross it with MYD88 and see if that will get us one step closer to Waldenstrom's. And just as a word of warning for all of you, if you're sitting minding your own business, working at your computer, and Ruben Carrasco comes around and asks if you're busy, careful what you say, because you will spend the entire rest of your afternoon trying to figure out what in the world the equivalent of a 6Q deletion is on the mouse. I say this from personal experience. So I'd really like to thank you for your support and for your attention. It's been a really long day, and this is a lot of technical stuff. But also, thank you for putting WM on the map. All this research, all the funding is possible because of you. And I really hope that everyone in this room can feel that and hear that. And WM is on the map today, not just because of researchers like myself or clinicians like Steve Treon. It's on the map because patients stood up and said, you know what, this is a real disease. There are people who are here. We can run clinical trials. We can work on this. And you guys made this happen too. So really, thank you so much. And I'd be happy to take any questions.